So again, with the Viking biology experiment, we had 3 official instruments as part of the biology package. And those were what?
The gas exchange.
Gas exchange.
Labeled release and the pyrolytic release.
Labeled release, pyrolytic release, and then there was also the GCMS. So the GCMS checked for what?
Organic material in the soil.
Okay, so we're looking for organic material. And why would scientists here on Earth send an instrument looking for organic material if they're interested in looking for life?
Because organic material is the stuff of life.
Yeah. It's basically, that's what we mean by living material. It's organic material. The main process that the labeled release experiment was checking for? Catabolism. Organisms break down complex materials to release energy and nutrients. So the labeled release experiment was essentially looking to see if there was anything in the soil that would break down labeled material. The opposite of that, the opposite of breaking things down, is anabolism, building things up. So if we expose the soil to radioactively labeled carbon dioxide and carbon monoxide, is there anything in the soil, either in response to light or some other energy source, it's going to take those simple materials and produce more complex organic material? And then the gas exchange was more general. It's just looking for any kind of metabolic processes that would influence the abundance of gases. So if there's life on Mars, according to very simplistic predictions ahead of time before the instruments are sent to Mars, the GCMS would find organic matter, because if there's no organic matter, we can't imagine as thought-constrained, earthbound investigators that without any organic matter, we can't imagine how living things would be there. The labeled release was looking for the breakdown of labeled material. So carbon dioxide released from catabolism, and the heated control should show no response. The pyrolytic release would show assimilation. Of CO2. The control should show no response. And the gas exchange should just see changes in gases if there's metabolism going on. And in the heated control, if there's no metabolism going on, we expect no response. So, in sending these instruments to Mars, it seems like a fairly clear-cut, simple test. You collect these soil samples, you put them in the different instruments, you heat sterilize some of the samples, you don't heat sterilize the others, you look for the expected response, metabolic responses in the experimental treatments, you make sure that there's no responses in the control, and if there is that difference between experimental and control, then as predicted here, and there's organic matter, then we have scientists jumping up and down and publishing papers and hoping to win the Nobel Prize. Clearly that hasn't happened because we're still talking about potential or not for life on Mars 4 decades later. So let's talk about that. Yeah, I'm going to skip over that. Okay. So if we look at the labeled release experiment first, here is the results of several runs of different soil samples fed through the labeled release. This is essentially, you take samples of the Martian soil, you expose them to the radioactively labeled organic material in the nutrient solution, and what is happening, what are the results we see in this over time? Sebastian.
That like whatever is put into the soil is like there is a reaction to it, and then like it kind of stabilizes a little bit over time.
So the radioactively labeled organic material, for it to show up in the carbon dioxide detector, something has to be breaking it down over time. And that then accumulates in the reaction vessel, and as more and more of the labeled material is broken down, more and more radioactive carbon dioxide shows up to be counted by the detector. Same thing for the samples at the Viking 2 site. A couple of samples run through, and so why would that level off over time? If there's life in the soil and it's breaking down this labeled material, why do you reach some plateau?
I guess 'cause there's only so much material that they could break down.
Yeah, you're only giving the critters in the soil, if that's the interpretation, you're only giving them X amount of chicken soup to break down, right? Once it's all broken down, the organisms aren't going to break down more of it because it's all broken down. So, we could very easily explain this response by the fact that there's something living in the soil, and we introduce the goodies, and the something living in the soil starts breaking down that material, and so it releases labeled carbon dioxide until it runs out of food, and then it doesn't continue to produce more labeled carbon dioxide. So that's pretty straightforward. What do you imagine was the response of the NASA scientists when they saw these results?
It was promising.
Looks very promising. Maybe it's time to get out the champagne. But this is just the experimental treatment. What should we look at next?
The control.
Okay, and why should we look at the control next? So it looks like we've got breakdown of material. It looks like the response you would get from, a metabolic breakdown by some microorganism. And then the question is, why should we then look at the control?
Because what they did was that they heated up everything so that if there were a what's it called,
Microbial activity?
Yeah. If they were in what they were testing, and if there was a change in it, then that means that it died, which meant that there was life in that.
Okay. So if instead of these untreated samples, we took samples and we heated them very hot and killed off microbes, what response should we see in the control situation when we feed radioactive food to it?
It wouldn't get broken down?
It wouldn't get broken down because we've killed off the bugs. There's no living bugs to break down the radioactively labeled food. And so we would expect that the sterilized soil, we'd add the nutrient broth, and there wouldn't be anything to break down the organic material, so therefore no carbon dioxide would be released to be detected by these detectors. So here are the results of the labeled release experiment when fed heat-treated, i.e. sterilized soil. Is this what you expect?
Yes, there's no activity.
Okay. So in this,
The second one is a little
you're heating up. Now let me blow this up a bit. Okay, so in the Viking 1 site, we have a control sample where the soil was heated up to 160 degrees Celsius before being introduced into the chamber. So, that's well above the boiling point. That's a temperature that would kill off microorganisms in soil samples here on the Earth, mostly. Except for maybe some very high heat-resistant spores that would not be metabolically active. And so you do that treatment, and lo and behold, there's essentially no response. There doesn't seem to be any breakdown of the material. Now in the Viking 2 site, there is, at least in the one sample, looks like there's a little bit of activity. But can you see what temperature the soil samples were heated to for the Viking 2 site? They're both heated to 50 degrees Celsius. So that's significantly below the boiling point, right? 50 degrees Celsius is fairly toasty. Your body temperatures, what's your body temperature on the Celsius scale?
Yeah, like 30.
37 about, okay? So, this is much warmer than mammalian body temperature. But clearly it's not as hot as the 160 degree treatment before. So why might we expect to find some response in these samples?
Because maybe more heat-resistant microbes were still able to survive.
Yeah, so 50 degrees C just might not be lethal for all of the bugs living in the soil. So you've killed off a lot of the bacteria, if that's the interpretation, but there's still enough around to metabolize a little bit. So again, as you might imagine, these labeled release results were initially and still by some people taken to be the clearest evidence of any of the experiments we sent to Mars that really showed a metabolic activity that disappeared when you heat sterilize the soil. So is that consistent? Is that evidence for life on Mars or not? For. Evidence for. Okay, so let's turn our attention then to the pyrolytic release experiment and the experimental results to begin with. Again, in this experiment, soil was exposed to carbon dioxide and carbon monoxide and allowed to react for a while. Then the initial carbon dioxide and carbon dioxide are blown off by helium being blown through the system. And then the soil is heated up to see if any compounds in the soil had taken up some of that carbon dioxide before it got blown away. So any kind of radioactivity coming off of the soil in that pyrolytic phase indicates that during the initial part of the experiment, there was something in the soil that took up carbon dioxide. So, what do we see here with the experimental results? Yeah.
There's still CO2 in the soil after it's heated.
Yep.
It shows that something did take in.
Okay. So all of these values for the different samples that were run, they're significantly greater than 0, which means there was, yes, some assimilation. So something took carbon dioxide, fixed it into more larger, more complex organic material that was then burned up in the pyrolytic phase to release carbon dioxide. So is this consistent with our predictions for life or not?
Yeah.
Yeah. Okay. So here's the control. Again, if the control killed off all biological activity and there was no anabolic metabolism going on, what response would you expect to see out of the system? You would not expect to see any in assimilation. That is, you expose the soil sample to the radioactive carbon dioxide, none of it gets taken up. The initial carbon dioxide gets blown away. Then when you heat up the soil, if no assimilation has taken place, you should not get any radioactive carbon dioxide coming off again. Do you see radioactive carbon dioxide coming off in the pyrolytic phase here? Yes. So even in the control, there's something going on to cause carbon dioxide to be assimilated into the soil. Now, if you look in detail, you can see that the soil sample that was heated up to 175 degrees Celsius, which is way above the boiling point, still has some assimilation going on, but it is reduced by about 50% over the values we had seen in the experimental samples. And heating to 90 degrees pretty much was similar to the experimental. So this is not showing a nice clear-cut prediction of untreated sample shows activity, treated sample shows no activity, right? So if this assimilation were due to metabolic processes and the heat treatment killed off any bacteria living in the soil, we would have expected no response in the control. So you can see, hopefully, that things are starting to look a little bit messier. It's not quite as clear-cut as with the labeled release experiment. And then finally, the gas exchange. I'll actually admit that when I've done this simulation in the past, I've not included the results from the gas exchange experiment because the gas exchange experiment is very messy. And what I presented here is really a simplification that really doesn't capture what the results technically really were. But The main point is that in the experimental treatment, we did see responses. Just putting it into qualitative terms, the really striking result out of the gas exchange experiment was a very rapid release of oxygen when the soil was exposed to nutrients. Is more rapid than any kind of response seen with terrestrial soil samples. And in fact, there really aren't any conditions where terrestrial soil samples with different kinds of microbes were exposed in this test container, and large amounts of carbon dioxide were rapidly given off. So this began to make the experimenters think that perhaps it's some kind of chemical process rather than a biological process. And like I say, the experiment was really messy. They then added more labeled material, and carbon dioxide and the oxygen that had been produced eventually declined over time. I tried to avoid some of that kind of complication from the experiment. Again, with the control, the results aren't as clear-cut, but the same kind of oxygen release was seen. So clearly the control is not providing no response, like we would have predicted from our simple predictions ahead of time. So complicated indeed. And then the final result from the GCMS. Essentially, what did we see with the GCMS? Was there organic matter detected in the Martian samples with the GCMS?
Apparently.
Yeah, there's just this one peak that shows up, it's methyl chloride. There's also some freon and other organochlorines that showed up, but they were thought to be contaminants from Earth. Because we use freon and things like that to clean instruments, getting them ready to go on these trips. So clearly, there's very little organic matter and nothing beyond the methyl chloride that is really detected at any great levels at all. You compare that to a soil sample from a desert here on the Earth, even a desert that's got what we consider on the Earth to be very low levels of organic matter. You see all sorts of organic matter that's available in the desert soils from the breakdown of different microorganisms and fungi and so forth living in the soil. If we took a soil sample from around here and put it into the GCMS, we would just be swamped with all of the organic matter that was in the soil sample. So GCMS is basically saying no organic matter in the Mars soil sample. Now, why is that a problem for our life interpretation?
Because if there's no organic material, then it could be life.
Right.
It's organic.
If there was something living in the soil that's doing all these metabolic reactions, then we would expect there would be organic matter. We would expect that we'd be able to detect that organic matter with the GCMS. So the scientists at the time looked at all these results together and said, well, there's clearly something interesting going on, but we don't think it's biology. We think it is chemistry. We think the chemistry of the Martian soil is different than the chemistry of soils here on Earth. In particular, we suspect that there are processes going on Mars that produce high levels of peroxides in the Martian soil. And those peroxides could explain the reactions we're seeing. Now, how many of you have had a cut and you pour hydrogen peroxide on the cut as an antiseptic? Okay. Basically, hydrogen peroxide is H2O2. It's kind of like a super oxygenated water. It's got this hydrogen, this double oxygen in it. And when it is exposed to organic matter, like the soil or bugs that you're hoping to sterilize out of your wound, it's going to oxidize that organic matter, break it down, and that's what was suggested, what was going on in the Martian soil. Jalen.
This is really random, but why is it called peroxide and not dioxide?
Yeah, well, carbon dioxide is a carbon that has two oxygens attached to it. The hydrogen peroxide, I'm not going to draw it still, with the right stereo arrangement, would be, with the two oxygens joined together. So this peroxide bond here is what makes the hydrogen peroxide so unstable and so reactive. And this oxygen to oxygen single bond between the oxygens is what allows the hydrogen peroxide to react with, or various kinds of organic matter to break that organic matter apart and stick a hydroxyl group on the fragments that are coming off. So the peroxide really refers to the type of bond here and the high reactivity. So that was really kind of depressing. Initial results get our hopes up. We get all excited. We see the label release experiment results, and it looks like we're finding evidence of life on Mars, and then things get complicated. And the vast majority of the scientists involved in the different experiments eventually, came to a consensus that, it's really chemistry and not biology, although the principal investigator for the label release experiment still argues to this day that the results from the label release experiment were actually evidence for life on Mars. as a little postscript, the problem with the Viking experiments is that, this is the first lander that we sent to Mars, and we tried to look for life right off the bat, and we had no idea, we didn't send along the instruments that would actually allow us to do the soil chemistry analysis to know whether, this is really the explanation or not. This is really just kind of, we're seeing all these weird results. If we take a look at soil on the Earth and we have soil that's got high levels of peroxide and expose it under these different conditions, we would see the same kinds of responses we saw on the Viking mission without having to refer to any kind of biology. You take sterile soil with hydrogen peroxide in it, and you expose it to the labeled release experiment. The hydrogen peroxide is going to break down the labeled food. Voila, we get the carbon dioxide released, and so forth. And that's why we also maybe expect to find no organic matter in the soil, because if, remember, the atmosphere on Mars is thin, there's no magnetic field, so the solar wind is constantly raining down on the Martian soil, which can easily lead to these peroxides being formed. And if there were anything trying to live in the top layers of the soil, presumably all of these peroxides and superoxides and other kinds of oxidizing materials would destroy that organic matter and essentially continue to sterilize the upper layers of the soil. That was the interpretation for 3 decades at least. But as a little postscript here, we have the Mars Polar Lander, the Phoenix mission, with run out of University of Arizona that went to the North Polar regions to actually look for, to dig for ice layers in the polar soil. Also had various instruments to assay the chemistry of the soils. And one of the important results that came out of the Phoenix mission was the detection of a compound called perchlorate in very high levels in the soil. Perchlorate is what was in was the oxidizer in the solid rocket boosters on the space shuttle when the space shuttle went up. So we know that perchlorate is a very highly oxidizing material. It could easily play the role that the peroxides were assumed to play in explaining the biology results from the Viking mission. But there's a couple of interesting points here. We know on the Earth there are microbes that live on perchlorate. They metabolize perchlorate as an energy source. So the fact that the perchlorate has high levels in the soil here could actually be an energy source for Martian microbes. And you could have basically a microbial metabolism that makes use of hydrogen peroxide and perchlorate to have kind of a weird life kind of microbial ecology. And the perchlorate plus organic matter, we could have organic matter in the soil, but if there's perchlorate in the soil as well, when we put that organic matter into the GCMS, the perchlorate would break down the organic matter before it was able to be detected by the GCMS. So there's been a lot of debate in the last decade, about 8 years or so, that whether or not the results from the GCMS really are to be trusted or given as much weight as they've been given. Could be that there was organic matter in the soil that Viking sampled, but the conditions under which it was assayed in the gas chromatograph, when you heat up that soil with the organic matter and the perchlorates, the organic matter could be broken down before the mass spec has any chance at all to detect it. So the negative result from the GCMS may actually not have been a negative result. It may have been a mistake. It could have been the perchlorate was masking the organic matter. And it really was that negative result from the GCMS, the fact that they didn't find organic matter, that really skewed or shifted the perception away from a biology interpretation of the Viking results to a chemistry interpretation.